Right, in this video, what we're going to do is we're going to look at what I call special inheritance, and there are two types of special inheritance. And we learned about both of these in baby bio, but there's a little twist to how we do them in this particular class. So Mendel chose these traits that obey these very specific characteristics. But what he did not realize is that there are traits that actually follow different rules. So, he chose characteristics that have a very definite dominant. The plant is either tall or it's short. The flowers are purple or they're white. There's no light purple. There's no medium-sized plants. But in reality, sometimes genetics works that way. So there are two types of inheritance where dominant has degree. The first is codominant alleles, where you have two dominant alleles and both are expressed on the side. So you actually see both of the alleles in the phenotype. The second type is incomplete dominance. And incomplete dominance happens when the recessive kind of peeks through a little bit. The dominant is trying to cover it up, but the recessive kind of shows through. And the result is that you get blending of inheritance. So let's look at what this looks like. The first I want to look at is the Andalusian fowl. I think this is the correct name for it. I can't really remember, but where if I take a black chicken and a white rooster and I cross them, all of the offspring are what I usually call with baby bio checkerboard, where you have both black and white feathers, right? Black, white, black, white, black, white, and they alternate. In this case, what you get is the black allele is a B, right? And the rules for how you assign these traits is both of the traits get a capital letter, right? So the white allele would get a B as well. And the problem is you've got to be able to mark B. I know sometimes online you'll see people doing B and W. I would encourage you not to change the letter, because if you change the letter, then that indicates you're actually changing traits. We preserve one letter per trait. So what I usually do is just make a little tick next to the B. So it's B for black and B prime for white. I've also correctly seen using the notation where they might use C for color, that then they use CB for black and CW white. That way you're keeping the same base letter so that you're indicating we're still doing the same trait here, right? But we're just marking them differently. So when we do this cross, the black chicken is BB. The white chicken is B prime, B prime. Sorry about the red on red there. So essentially, here's our white chicken on the top here. Here's our black chicken on the side, right? And all of the offspring are B prime. So this is how this Punnett square works. Okay. So this BB prime, this is the black and white offspring that we're seeing, that checkerboard chicken. Now, what characterizes this is you'll notice there are three different genotypes, and each one has its own phenotype. So the beautiful part about this is that the genotypic and the phenotypic ratios will always be the same with this type of inheritance, because BB is black, B prime is white, and then if you have the one of each, the heterozygote B prime, it's checkerboard. So genotypic ratio and phenotypic ratio will always be the same. So that's lovely to do it. And you can do we're going to do some examples in class, but I just wanted you to have a chance to do this. All right, now the next kind, you can use the notation of CR and CW for color red, color white. What a working geneticist does, this is 4 o'clock plants, is they use the standard notation here, because there is a definite dominant and a definite recessive. The problem is when you use the standard notation, you've got to remember that the heterozygote is the pink or lavender variation. Okay, so if you take, for example, in this particular flower, a pink plant or a red plant, I think they're using R for red, and you cross it with a white plant, all of the offspring turn out this light pink or lavender. They're calling it lavender, I don't know why, but pink flower color. So again, the cross is just your standard, whoops, sorry, hold on. right? So here's our cross, right? So you can see really easily that all of the offspring here would end up with the heterozygous genotype, right? But that genotype is not red, right? What's happening here is that the white is showing through a little bit and it's diluting the red down to this pink color that you're seeing. Okay. So if we then cross two of these individuals, these heterozygotes, then you would see the 1 to 2 to 1 ratio of, in this case, they're saying purple, lavender, and white. In the forecock plants, it's red, pink, and white. Okay. So that's incomplete dominance. But again, notice the difference here between this and codominance is really that you see a blending of inheritance instead of seeing both traits side by side. Before we saw the black feather and the white feather. Here we're seeing a blend of those two colors together to give us that lighter intermediate genotype. But also, just like in complete or in codominance, you still have one genotype for each phenotype, right? So there's the genotypic ratio here and the phenotypic ratio are always going to be the same. So that's a nice feature about this type of inheritance as well. But that's both of these, and we'll do some examples in class.